Hello student, this is Shumona from SIMMI Foundation. Today we are here to learn about an interesting topic named different types of antibodies. In our previous video, we already learned about what is antibody and the structure of antibody. So here we can recall that an antibody is a Y-shaped molecule with two identical antigen binding sites, one at the tip of each arm of Y. Because of their two antigen binding sites, they are described as bivalent. And it consists of two identical light chains and two identical heavy chains. In mammals, there are 5 classes of antibody or immunoglobulins, that is IgA, IgD, IgE, IgG and IgM, each of which have their own class of heavy chain alpha, gamma, delta and epsilon. So now we will learn about the 5 antigens in separately. So here we will start with IgG. IgG is the most abundant antibody in serum, constituting about 80% of the total serum antibody. They has a half-life of 7 to 23 days. IgG is a monomer and has two epitope binding sites. This is the only class of antibodies that can cross the placenta and enter the fetal circulation. Now we have to know the function of IgG. Immunity to newborn, neutralization of toxins. IgG3 binds to Fc receptor by phagocytosis. These maternal antibodies will protect the newborn until its immune system is sufficiently mature to produce antibodies on its own. Now we will learn about the antibody type IgM. IgM is the first class of antibody made by a developing B cells. It accounts for 30% of the total serum antibodies. They has a half-life of about 5 days. Most of the IgM are pentamer and has 10 epitope binding sites. Some are monomer. It is the first immunoglobulin class produced in a primary response to antigen. Now we will learn about the function of IgM. Activation of classical pathway, defense against multivalent antigens, act as opsonin. Now we have to learn about the antibody type IgA. IgA constitutes of approximately 6% of serum antibodies. They has a half-life of approximately 5 days. IgA is a dimer and has 4 epitope binding sites. They are found mainly in body secretions such as saliva, mucus, tears, colostrum and milk. And the function of it as a secretory antibody, effective against virus that causing influenza, producing to infant gut. And we have to know about the antibody type IgD. It constitutes 0.2% of serum antibodies. IgD is a monomer and has 2 epitope binding sites. These class antibodies are found on the surface of B lymphocytes. IgD and IgM is the major membrane bounding antibody expressed by mature B cells. Now we have to know about the function of IgD is B cell activation, which is with IgM, and act as a receptor for antigen binding. And the last we will learn about the antibody type IgE. IgE is a very low concentration in blood, which is 17 to 450 nanogram per millimeter. It contains small percentage of lymphocyte, and the function of it are responsible for immediate hypersensitivity, bind to Fc receptor on basophil and mast cells, release of substances like histamine, vasoactive theaters. So student, in this video we already learn about the five types of antibody and their functions. So for today that's all, and thank you.